It's time. Okay, we have way too much stuff to cover in order to get through the syllabus in class. But there's always those outside of class moments that are so special that you can look at the new audio files that I have not made yet, but plan to make tomorrow. I'm going to make an audio file on the Operon and cap. So cap regulation will be the first one, and that'll be about, it has about half the material that we'll cover today. And then I'll make a final new audio file on the tryptophan Operon, which is about half of that. So that's a really short one. I'll take you about 30 minutes to go through. So, because most people don't take this course by actually sitting in the seat, it shouldn't make much difference to people. Everything in that syllabus, all the way through the trip Operon and attenuation, will be on the exam, which opens up Saturday morning at 8 a.m. I doubt if anybody will be taking it then, but some people take it on Saturday. I will give a review session that I'll try to stream in the, and try to record if I can get my act together. Saturday afternoon in the microbiology cell science main seminar room from 1 to 3. It's a good opportunity to ask me questions, to have me go over something. I can go over stuff that I don't get to go over in class maybe. You can divide up in groups. One of the TAs will be there, and she has a whole bunch of flashcards she prepared last year, and she just loves to torture study groups with those things. So it's a good chance to form a sort of an informal study group if you like, or you don't have to. Okay, so let's get going today. We got a lot to cover, but we should be able to get through it. The Operon. And last time we started into this PowerPoint, and there's sort of a prologue where we go and get more information on the CTD of the alpha subunits, because those are key regulatory components of the holoenzyme. They're actually a part of the core, but they don't really function until they're part of the holoenzyme. And so what are the main points that you should remember about the CTD of the alpha subunit? First, there are two alpha subunits, right? One makes direct contact with beta prime, and the other one with beta primarily. They can't be contacting exactly the same space because there are two of them. So you have two of them, and they have an N-terminal domain that's connected to the core. They have a flexible linker that's connected to either to DNA directly at the UP element, or they make contact with a transcriptional control protein. And the only one we're going to talk about is cap, and that will be recorded in the audio file. But they make contact with cap, and so it binds the UP element. I'm fond of asking, how is that contact made? Which groove of the DNA is used? It's the first example out of three that we'll talk about that interacts with the minor groove of DNA. You understand major and minor grooves of a double helix? If you don't, you need to maybe Google DNA structure, and you'll see there's kind of a groove that goes around the outside of the double helix as they twist around each other. There's one that's a little wider than the others, like two candy stripes that go around the same direction. One's a little wider and one's a little bit narrow. Predominant interactions occur in the major groove. There are only few examples of interactions where it can actually read bases in the minor groove. And this is one of those examples, and it uses not a helix turn helix, but this helix hairpin helix. And the last thing about the CTD of the alpha subunit is, how does it stimulate transcription? Is it some fancy mechanism that involves bending of the DNA? And it may involve bending of the DNA, but it's not to melt the promoter. It may be bending the DNA to make better contact with the alpha subunits of the core. So it does definitely bend the DNA, and when it interacts with CAP. I think I've got the two things mixed up here. CAP bends the DNA. The CTDs, the alpha subunits, don't bend the DNA. Okay, how does it work? Simply by increasing the affinity of the polymerase with the promoter. Makes it stick to the promoter better, and that results in greater activity. And a lot of ways that you can do that. One is that it interacts directly with the DNA. Another, it interacts with a sticky protein that's already there. Okay, so let's move right to the Operon proper. We'll start with definitions. What you see here is structural genes. In fact, this will be polycistronic. This is usually a biochemical pathway of some sort, although it could be for a structural RNA, right? Like the ribosomal RNAs, purely structural RNAs, they don't make enzymes, but a lot of the ribosomal structural RNAs are all lined up in a row, polycistronic. Protein coding, they'll be all lined up in a row for entire pathway sometimes. Other times not, but it's the predominant organization pattern in bacteria that you have one promoter and you make a whole series of genes that are that need the same kind of regulation. You save space, you simplify regulation by having one promoter make all of it at the same time. Okay, so this is what we call the operon. It'll be the promoter and the transcript. There's usually a second part that in some textbooks is called a part of the operon also, and that's these regulatory genes, whether it's a positive acting transcription factor that's needed to make the operon promoter work, or it could be a repressor that controls the promoter of the operon. This regulatory gene is sometimes located right next to the operon. Other times it could be located all the way across the genome. For purposes of this course, we will say this regulatory component is not by definition a part of the operon proper. It's the regulatory component of the operon. Here's the definition. It's a complete regulatory system, includes structural genes encoding either proteins or RNAs. The promoters and cis elements of that polycistronic message, the genes encoding specific regulatory proteins, which in turn control the operon, activators and or repressors, form a separate unit, are not considered part of the operon. That's not a good, okay. Whew, thought the world had ended. Cis elements. These are promoter elements, sequences on the DNA that things bind, regulatory proteins bind. A cis element could also be a part of a promoter where RNA polymerase or the sigma subunit would bind. So it's a fancy term for something that's pretty simple. Here's a very specific term, operator. It's only used in one context properly. It is by definition the binding site on the DNA, and since it's regulating an operon, it would be in the promoter where a repressor binds. Not a positive acting factor, not the polymerase. An operator, by definition, only means one thing. It's the site on the DNA that the repressor binds. If there's no repressor controlling this operon, it's controlled by a positive acting factor, there will not be an operator there. There can be more than one operator in the promoter. We'll see these kinds of possibilities later. Transacting factors. Trans means it is made someplace and then moves to a second location to act. It doesn't have to act at the very location where it is physically located. A cis element, of course, acts where it is located only. It only controls the promoter that it's embedded in. It can't control a promoter in another halfway across the genome. This is kind of an obvious distinction. Okay. Regulation can be negative or positive. It seems like a very simple concept, but it's easy to get kind of balled up with it. Negative or positive. If it's negatively regulated, just think about that. That means the default condition is it's always on. And you, to regulate it, you shut it down. It always involves a repressor. That's the giveaway. If there's a repressor controlling the operon, it's under negative regulation. Positive regulation. It means you have to contribute something to make the promoter work. It has a lousy promoter. It needs supplementation. It needs help to make the promoter functional. A positive acting protein must be a part of it. So negative always means there's a repressor involved. A positively controlled operon always has a transcription factor that stimulates transcription. Okay, let's talk about repressors. How do bacteria use proteins that interfere or inhibit gene expression? If you were doing it, how would you do it? Now, you have a brain. E. coli does not. What's the simplest way you can think of? Just get in the way. Oh, excuse me, no, I'm not moving. I'm a disruptor. That commercial, that guy's under the seat looking for a cell phone. Chaos is there. Repressors get in the way simply by just sitting down and getting in the way. The polymerase goes, this is my seat, I got the ticket, it says this is the consensus right there, and you're going, I'm sorry, it's my consensus too. And now it's just a question of where do you put that pesky repressor? You can put it right next to where the polymerase should be, so there's not enough room for the polymerase to actually completely get its haunches down onto the promoter. So sort of behind it, overlapping it, or you can put it in front of where the RNA polymerase binds so that it sits down and look, it's got this, it's like facing a brick wall. It can't see the screen. Please take your hat off. I can't see the screen. In fact, it'd be great if you could go get yourself a drink at the concession stand. Location, location. And it's so, there's no enzymatic activity involved in making a repressor. You just make it so it binds DNA at an inconvenient position. That's how you make a repressor. The LEX-A repressor, it doesn't do anything but just sit down and get in the way. Of course, life can't be that simple, right? The lac repressor, which is what you've all learned to fear and dread, it's the reason why, only reason you hesitated in the slightest from signing up for this course, because you figured there might be a little discussion of the lac repressor. That thing's a little more complicated, and you have no idea. You think you do, you don't. You'll see. You were spared. Okay, but I will not spare you. You'll get all the gory details right here. So promoter binds the -10, the -45, An operator can overlap it in front, behind it, all sorts of places. That just, a repressor sits there and I'm sorry, polymerase, there's just no more room here. Oh, there's a repressor. Oh, now that's accommodating, isn't it? This is what the lac repressor does. Oh, you didn't know that, did you? The lac repressor binds and overlaps the promoter, but it hangs on underneath. You get in the car, there's somebody in here with me. You look out, look underneath, and oh, it's a lac repressor. The lac repressor is hanging on underneath, and it is making contact with the RNA polymerase itself. In fact, it is holding the polymerase down and keeps it from floating up, because this is such a lousy promoter that the RNA polymerase doesn't have much attraction or affinity for this. It has to have the assistance of the repressor. The world's gone crazy, right? Repressor is now bringing in, increasing the affinity of the polymerase for the promoter. There must be other things going on, and I assure you there are. The lac has affinity for RNA polymerase. In fact, the lac repressor makes it so that RNA polymerase has 100-fold greater affinity for the lac promoter because the repressor is there. Wow, didn't know that. More about that later. That's just to kind of get your juices going. All these years, they were keeping this from us. They were. They never even whispered that to you, did they? Nope. There you go. Positive regulation. This is where it's a lousy promoter, and it won't work unless you add something. A transacting factor that activates. A protein is required. It's off by default because it's such a lousy promoter. Uh-oh, what does this say? Exam 3 alert? I didn't mean for that to get there. There's a question related to what this means. It'll test your understanding of it. So let's look at it, and you'll probably realize things you didn't realize before. You notice it's got it's got several kinds of things going on here. Negative control and positive control. Everything in this column is negative control. So what do you think you're going to see? A repressor in both of those panels. There's an inactive repressor and an active repressor. But the common theme is, if it's negative control, a repressor's got to be involved someplace. Okay, let's look at positive control. Again, there's a controlling protein, but it's an activator. So right off the bat, if you looked at a column like this and I covered up a label and said, what's this thing? You go, negative control's got to be a repressor that's represented there in that diagram. Let's look at other things. So you have a repressor. How can you ever induce activity when your regulation is negative? Look, repressed, induced. Repressed, induced. That is what's common, except down here they've turned it around. Induced, repressed. So you can talk about induction even though you have negative control. You can talk about repression even though you have positive control. Now it begins to kind of blur your states of consciousness. You thought it was simple, now it's going to take some studying just a little bit to make sure you're not all balled up when I throw a question at you, right? So, obviously, in each panel, you're going from either repressed to induced or from induced to repressed. That's what each panel has in common. In this column, you're doing it with a repressor. In this column, you're doing that same thing, on-off switch, with an activator. Notice induction. That's the row. Everything up here means it's not active at first, and now it becomes active. You can induce using negative control. You can induce with positive control. So how would you do it with negative control? You would start with a repressor that represses. And then you can have a small signal molecule which inactivates the repressor. And that would result in de-repression, which is the same as saying induction. So you can induce by killing the activity of the repression. Destroy repression and you have induced. So look, under positive control, you do it the opposite way. You start with a positive activator. That's required, right? But you make it so it's inactive. So transcription is repressed. And then your small molecule that controls it renders the activator active. In each case, you have a protein and a small signal molecule. They act with opposite effect. But it's the same general scheme. You can see down here, repression. So you start with an inactive repressor and you make it active with a small molecule. Here, positive control is going to be repression, so you start with an active activator and your small controlling molecule deactivates the activator. So there's a lot in that diagram that is useful. It gives you kind of an overview of what we're talking about. But you see, it's all very logical. Okay, let's dive into, I think this is where we dive into the lac repressor. You know, it makes lactose, one of the signatures of our phylum, Mammalia. Turns out to be one of the first genes that the tailless ape figured out, how lactose is metabolized in a gut bacteria. So here you see the promoter of the lac operon. Here you see, actually this is not the promoter of the lac operon, this is the promoter of the repressor. It happens to be located right next to the official operon. This is the promoter for the lac operon from here over. This is the regulatory gene that makes a repressor that controls expression of the operon. Notice this particular promoter has an operator built into it. This repressor is going to be binding there. Notice it's polycistronic. It makes 3 different enzymes: galactosidase, permease, transacetylase, like Z, Y, and A. The operon, the regulatory gene. So here you see the more detail of the lac promoter. Let's talk about the repressor protein. It acts as a tetramer. That's the first thing you need to know. That's kind of odd because DNA binding proteins generally always bind, not always, but 95% of the time, they bind as a dimer. Here you've got a tetramer. And the implication you'll see, the reason that is so is because really you have 2 dimers joined together, so this thing called the lac repressor can simultaneously bind 2 operators at the same time. Whoa, nobody told you that either, did they? That's what lac repressor does, binds 2 at the same time, otherwise it won't work. Uh-oh. It's getting a little more complicated, but not much. You'll see. So first of all, before I reveal that mystery as to why it needs to grab at two places, let's talk about the expression of this poor repressor, the lac repressor. Poor thing, it's probably the least expressed gene in E. coli. Why is that so? Which of the following is not a reason for the low expression of the lac repressor? Well, here are two reasons it is so lousy. One is it has a horrible promoter. I looked at it before I came over here. I cannot see a -10 tata at all. It's just a horrible promoter. That's one thing. And the second reason is it's a horrible messenger RNA. That thing does not even have a Shando-Garno. There's no place for the ribosome to bind. And furthermore, it doesn't start with AUG. It starts with that alternative start codon used in E. coli. Anybody know what that is? It's, yeah, GUG. It says so right there behind me, doesn't it? You guys are sharp. It gets smaller and smaller every year. Wonder why that is. Okay, GUG, I can't keep any secrets. So they're the two reasons. A lousy promoter, and it starts immediately with the start codon with no Shando-Garno, and it's using this alternative start, GUG instead of AUG. Okay, so back now, we've sort of learned a little bit about the lac repressor. Let's learn more about the control, what we see. How is this lac operon regulated? We know it's controlled by small molecules, and this is sort of a general statement here. Small molecules that induce an operon for a catabolic pathway are identical or related to one of the substrates of the enzymes that it makes. What in the world did I just say? What is catabolic? It's the breakdown, degradation. If you want to use a sugar as a nutrient, as a food source or energy source, you need to break it down into its constitutive parts. And so breakdown pathways that go out into the world and feed on the steamed cauliflower and the broccoli and then digest it to break it down, those pathways are induced by the presence of the food itself. Cauliflower turns my stomach, well no, it turns my stomach on. Lactose, or one of the breakdown products of lactose, induces the lac operon. It says, lactose in the neighborhood. It's time to gear up the polycistronic operon so we can degrade lactose as fast as we can. Catabolic enzymes. What's the opposite of catabolic? Anabolic, but they synthesize something. If you're going to synthesize something, you usually are controlled by the end product. You're measuring, do I have enough of the end product? If I don't, I need to make some more. That's logical. If you're going to destroy something, chew it up, your signal is, well, is the substrate around anywhere? So you're looking for the first step in the pathway. Okay. So beta-galactosides are substrates for the enzymes encoded by LAC-Z, A, and Y. In fact, you'll see there's a LAC-Z is beta-galactosidase. There happens to be another one in this E. coli that works at a very low level all the time. So it's producing the breakdown product of lactose at low levels if there's any lactose around. And that breakdown product, the beta-galactoside, the breakdown product of lactose, is what induces the real operon. The absence of beta-galactoside, the lac operon, is expressed only at a few, 5 proteins per cell. This enzyme is there from another promoter, and this one's a little bit leaky also. So you have 5 enzymes per cell is pretty low, but it's enough to create a beta-galactoside breakdown product of lactose if any lactose happens to be around. Here you see the expression properties. You add inducer, that would be a beta-galactoside. You can order a synthetic beta-galactoside from Sigma Chemical Company, quite cheap, and it's used to induce plasmid protein expression because they use the lac promoter to induce all sorts of expression vectors in the lab. You put in IPTG, and I can't remember what it stands for, but it's a beta-galactoside that strongly induces the lac promoter. But so an inducer, it could be the natural beta-galactoside, and this is the RNA. You'll see in minutes, you've got full induction in 6 minutes. But look at the enzyme. The enzyme lags a couple of minutes, and then you've got the full level. What's the stability, the half-life of a typical messenger RNA in a bacteria? On average, it's about 1.5 minutes. Yeah, it's pretty quick. So you can quickly up and down regulate things. The same systems, in other words, you have a protein that either represses or activates, and it's controlled by a small signal molecule. That strategy controls both biosynthesis pathways and catabolic pathways, that general approach to regulation. That's all this slide says. What's an example of a biosynthesis pathway that we're going to look at? Tryptophan. It'll be the synthesis of tryptophan. The other promoters, arabinose, lactose, those are going to be catabolic pathways, breaking down those sugars. Good question. Would you expect eukaryotes to use the same mechanism to repress transcription? And to repress transcription, you have a protein that just sits down on the DNA and gets in the way. Do you think a eukaryote would use that? I mean, we think of repressors and activators. Surely we do that, don't we? Nematodes do it, or algae. Somebody does it, right? Eukaryotes don't do it that way. Uh-oh, how in the world could they possibly do it then? Eukaryotes have repressors all over their entire genome all the time. Histones. They have such large genomes, they have to keep it under wraps all the time. It's not like they had a promoter open waiting to be repressed. They're totally repressed all the time. They can't use this strategy. Okay. We're getting a little more into detail here. Here's the lambda repressor, and I have no idea why they draw it looking like that. I don't even know what that is. It looks like some larva that lives under a pine tree and becomes a locust every 17 years. I don't know what that is. It's a big pink knobby blob that they're calling the lac repressor. So we'll go with it. Inducer functions by converting the repressor into a form that has lost its affinity for the operator. That's how the repressor works. It's controlled by a small molecule, IPTG or beta-galactoside, that renders it inactive to repress. And it does so by making it so it cannot bind to the operator on the DNA. 2 binding sites. I didn't get all of them. The book calls them binding sites. No. They are domains, they are protein domains. Binding sites refers to elements on the promoter, on the DNA, where the repressor comes and binds. That's its binding site. It has within it a region, a domain that is specialized for DNA binding. So a domain refers to the location, the folds, the alpha helices in the protein itself that's responsible for going to the binding site. So the book has already confused the terminology, so it's hard to talk about it. So I have tried to tease the two apart. That should be repressor has two binding domains. One domain binds the inducer, the other binds the operator DNA. So when I go back and edit that, I'll say it has two binding domains because it's within the protein, they are domains. Repressors inactivated by the allosteric interaction. In other words, when it binds the beta-galactosidase, it changes the conformation of the LAC repressor so it can no longer bind the operator. Mutations in the operator, that's the binding site, causes constitutive expression of the polycistronic message. I'm explaining figures from the book. And so that makes sense. If you can't bind the repressor, then there's no way to shut this thing down. So it's said to be constitutive, meaning unregulated, always on. Constitutive in the context of molecular biology means that. Mutations are cis-acting, which is, they've made something simple complicated. Cis-acting means if you mutate this promoter and you have a second copy of the promoter that's not mutated in the cell, the mutation's only going to shut down this one, not the other one. Duh. Mutations in the repressor gene is called LAC-I. LAC-I is the name of the repressor. It's the name of the gene. Transacting, in other words, if you mutate the gene that makes this protein called a repressor, then wherever that protein represses at every promoter in the cell, you're going to affect its activity. It's transacting. So again, that's not a complicated concept. They just make it complicated with terminology. Mutations that totally eliminate LAC function, if you totally destroy the LAC repressor, of course it's going to be constitutively expressed everywhere. The same phenotype you'd get if you mutated the binding site, the operator on the DNA. You couldn't bind, it's going to be constitutive everywhere. Mutations in the DNA binding domain. I've changed the term. I used to say binding site. Mutations in the DNA binding domain of the protein, of the repressor, are constitutive because the repressor cannot bind to the operator. That makes sense? Mutation in the inducer binding site of the repressor protein make it so that it cannot be induced. In other words, the repressor always represses. You can't de-repress it because that's what the beta-galactoside does in the case of the LAC operon. The active repressor is a tetramer of identical subunits, only 10 tetramers per cell. Yikes. That's why it's so poorly expressed, you don't need much. I mean, that's half the number of Paul 3, right? They're about 10 to 20. It's very low expressed. And here's the odd thing. When you have a mutant, if you have two copies of the repressor gene, cells don't, but if you did, you would find that if you mutated one of them, that you would get neither repressor working anymore. Why would you think that would be there? That's called a dominant mutation. You got a wild type gene, represses perfectly well, you got one that's all messed up, and if you have them both in the same cell, the one that's all messed up is going to mess up everybody. That's because it works as a tetramer. There'll always be a rotten apple among the four. At least one. And that's why it's dominant. Because all four have to work before the LAC repressor can actually repress. And you guys don't know why yet, but remember it's related to the requirement that it bind two sites simultaneously, two operators simultaneously. At each operator, you've got two monomers, two here, and they both have to be functional, and over here, two monomers. All four have to be working in order to shut down the lac operon. And that's what the rest of these slides say. You have a mutation DNA binding domain, and then you have a wild type copy there. The mixture, one out of the four, is enough to destroy its ability to control transcription. Okay, so we're kind of going in and out of the details of the lac operon. Now we're taking a step back and looking at repressors generally. Where would you put them? And I've alluded to this a little bit. Location, location. This is showing that operators can be at several different locations. Here it's probably illustrated better, the dark shaded areas showing you can put it in front, behind, overlapping, all sorts of configurations. You don't need to memorize this at all. Just realize any place that gets in the way of the polymerase is someplace you're likely to find an operator. Here's a question that the book poses, and this was all done about 25 years ago, but the initial questions were, how does this small molecule regulate things? Does the tetramer have to come off of the DNA before it's subject to regulation? Or can it be regulated by the small molecule while it's still bound to the DNA? And of course the right answer, and I guess that little arrow I put there doesn't work. I probably didn't upload the latest version, but there's an arrow that points to this being the correct answer. Experimental observation in vitro, IPTG, that's the synthetic beta-galactoside, causes immediate release from repression. The half-life of the repressor DNA complex is 15 minutes. So if it slowly is going on and off, and it can only inactivate the repressor when it's off, you would not expect to have immediate repression. The fact that you have immediate repression strongly suggests that the small molecule, which is IPTG, can actually bind to the tetramer while the tetramer is actually at the promoter and cause it to release. What does a typical operator look like? They are usually inverted repeats, right? Not necessarily completely adjacent, but very close. And what you'll see is there won't be a perfect, see the T's here, the A's there. They're not perfect, but it's enough binding so that you can get a dimer to bind. In fact, it's kind of a lousy site if you had a monomer, if you required a monomer to bind, because it's full of imperfections as a perfect palindrome, as a perfect inverted repeat. In fact, it requires the two to bind together. Why is nature doing that? Why did it, it's so easy to make that a perfect binding site. Why is it deliberately flawed like this? And this is the way most genes are regulated, with these inverted repeats, with these all messed up, imperfect inverted repeats. They're imperfect. It's deliberately so the monomer doesn't bind with very high affinity. But if you have two of them, they stabilize each other. As one lifts off, it's being held down by the other one because they're a dimer. And the other one goes down, okay, I'm coming, whoop, and then that one goes off. It's like this. But it always remains stuck. They synergize. Synergy is exploited. But you go, wow, why did you have to go to all that trouble? Look, that's only 5 or 6 bases. That's only one helical surface of the double helix. That's all you can build a protein helix to make contact with. It's very hard to build a protein helix that can wrap around like a big spring. It can only read at maximum 5 bases. If it's 10 all the way around, it can have a little bit of curvature and reach 6 off the odds and ends and loops and stuff. But that's because the DNA recognition proteins do it through an alpha helix. And the alpha helix of the protein can only sit down on one helical face of the DNA at a time. So why is that important? Well, that means that if you're going to build a very specific regulatory sequence, you've only got 6 base pairs to do it with. And you got 4.2 million base pairs in the genome. Just think how many times by serendipity, by accident, you're going to build a powerful transcription regulatory site by accident. This requires now that the binding site be much more complex in terms of probability that it will appear. You've built in complexity just by repeating. It works. That's why you have dimers. You seldom have a monomer. And if you do have a monomer control protein, it usually has multiple DNA binding domains embedded within it. Just for that reason. To deliberately make the binding site complicated so you won't get regulation at crazy places throughout the genome by accident. I bet light bulbs are going off all over the place. Why didn't somebody explain that to freshmen? Think of all the stuff you could have done. I don't know. So, I think that's a pretty neat, powerful concept, actually. That's the 2 monomers. They're joined together. This is the DNA binding domain, and then you'll have usually some kind of oligomerization domain that holds the 2 together. Okay. Oh, this makes you done this. You don't even want to look at this, right? Just turn your head away. This association constant, that's what the A stands for. It'll just look, it's kind of logical. The repressor DNA complex. It's the ratio of what's in a complex bound to how much is free, the free repressor and the unbound DNA. You just figure out the molarity of each of those things, and that ends up being the association constant. Well, if you rearrange it, you can actually solve it, and you can solve it for the lac operon, and if you do, you'll notice that the concentration of DNA is very high. It makes that number quite small because it's a ratio. Anyway, it comes out to be 10 to the minus 4th. If you change that to a percentage, that means, just simple make that a percentage, it's 0.01% of lac repressor is bound to random DNA. Only about 10 repressor molecules per cell, so there's no free protein. All but 0.1% of repressor is bound. Wait a minute, that said random DNA. That's right. That lac repressor just loves DNA. Even non-operator DNA. You never see it floating around. I mean, what is 0.1% of 10 molecules? You can't do it. You can't split up a tetramer into a percentage that low. So that means that essentially every one of them is always bound to DNA. Whether it's operator DNA, which is where it's supposed to be, right? Or non-operator DNA, which is the whole rest of the genome. Nobody told you that either. So that's the take home from this. Just figuring out the K association, it gives you a prediction that there's no free repressor. It's all bound. This is the last slide with numbers on it. So I'll tell you when you can wake up. Size of genome. Okay, so this is, if you have a very large genome and you only have one operator, the more random DNA you have, the harder it is for your repressor to find that random piece of DNA, because it just loves any old kind of DNA, right? So that means the specificity of the protein counters the effect of mass. Therefore, the amount of protein needed increases as the size of the genome and decreases with binding specificity. Let's talk about binding specificity here. It's the ratio of specific versus non-specific binding. So let's take a look at, first of all, these are the K association constants. So you have operator, repressor at 2 times 10 to the 13th, repressor from plus inducer. This binding affinity decreases, you can see, from 13 to 10, it decreases 1,000-fold. It doesn't want to bind the operator. 1,000-fold less. It has 1,000-fold less affinity for operator in the presence of a beta-galactoside, or IPTG. You go, yay, right? It lets go, floats around. But look at the other component here. Non-operator DNA. It has 10 to the 6. That's a big difference. That's a 10 million-fold difference in binding specificity for operator plus non-DNA. So you say, well, why does it float around? It just loves operator. A 10 million-fold difference in their affinities, in their association constants. But look over here, repressor plus inducer. This is inducer. That means when the repressor is in the inactive state, it still has a pretty good affinity for DNA. That's an only, 10 to the 4th difference. It greatly reduces its affinity for the operator, but not as much reduces its affinity for stupid DNA, non-operator DNA. That 10 to the 4th is insufficient to capture repressor. Repressor with 4 times 10 to the 6 DNA sites. This would be the number of different sites in the rest of the genome. Okay, so you can wake up now. No more numbers. I'm sorry I had to do that, but we're through with that now. Now there's no reason not to sign up for the course. Wait, this is a stupid professor, keep going back. Now let's look at pretty pictures. Yay, right? Pretty pictures, and I'm going to put labels and ask you where the arrows point. This, believe it or not, is a single monomer of the lac repressor. You got to get 4 of these joined together to make it functional. So let's look at the parts of the monomer first of all. You have at the N terminus, you have what looks like a helix turned helix. In fact, that is what it is, a helix turned helix. It's a DNA binding domain. This interacts with 1/2 of the operator, right? One of those arrows in the inverted repeat, and it interacts in the major groove. A helix turned helix interacts with major groove. Helix hairpin helix interacts with minor groove. So far so good. Then, so that's 3 helices taken up here. 1, 2, 3. And then the 4th helix is called the hinge helix. I'd love to ask you about this guy, because that is a very unstable, poor excuse of a protein alpha helix. It's just really short and it's not very stable. The only way you can stabilize that and make it actually be a protein alpha helix is to have it sitting in the minor groove. So this thing is sort of like a tripwire. If you can pull that thing out of the minor groove, by rearranging the conformation down here because you've bound an inducer. If you can yank that out of the minor groove, then it's like the limp went wrist. The wrist went limp. And this thing now, not so much interested in binding in the major groove anymore. It is, the heck with it. So it has sort of a trigger mechanism here involving a very unstable hinge helix that is only a helix when it's sitting in the minor groove. This is sitting in the major groove adjacent to the DNA. So as you guessed it, this is where the inducer binds. Now, on a monomer, you can't really see how that would make much difference, but we'll talk about it in a minute when we put the whole thing together as a tetramer. So what else do you have here? You have these two globular domains. We'll call this a globular domain, a cluster of helices and beta sheets. In between, you have the inducer, the IPTG binding site. And then at the end, you have this interesting thing here. This is another alpha helix. This is going to be where the dimers are held together at their tails. Two dimers are going to be held together at their tails by the intertwining of these alpha helices at the C terminus in what's called a leucine zipper, or as I like to say, a leucine lipper. Inducer binding site and regions responsible for dimerization are located in the core. Questions I love to ask you. How are the two monomers held together? They are held together by interactions between these two globular core domains. And then I'll ask you, well, how are the two dimers held together to make a tetramer? And that's by the intertwining of the C-terminal helix, or helices, to make a 4-member leucine zipper. So monomers make a dimer, make a tetramer. Here you see a dimer, and they're held together by core-core interactions, and you've got two members of what is eventually going to be a leucine zipper down at the C terminus. And now you've got two dimers coming together, and they're held together by a 4-member leucine zipper. This is kind of looking at it on end. If you had 4 springs and you had them in a bundle with a rubber band, and then I pointed them at you like that, that is what that's the view you're getting from that. And the springs slowly coil around each other, very slightly. That's a leucine zipper. You can have 2 helices coiling around each other very slowly to make a 2-member leucine zipper. You can have a 3-member leucine zipper or a 4-member leucine zipper. And here you see, this is the DNA. This is one operator site. This is one monomer binding. This is the other monomer binding. This is the second operator. One monomer binding, the second monomer binding. And look how this DNA seems to be in some kind of crazy pattern. This is really going like this, forming a big loop and coming down like that. That is the reason it has to bind 2 at once, so it can make a big loop of DNA. That loop of DNA shuts down transcription because the RNA polymerase, sigma, cannot melt the promoter. You know, if you had a rope and you clamped in front and behind the RNA polymerase and you tried to open up the strands, if I'm clamping the strands tightly, it'll be very difficult to melt the promoter or to open up the rope. But if I let go on one side, I can open up. Let go on the other side, I can still unravel the 2 strands. That is how the lac repressor represses. It lures the RNA polymerase to the promoter and then clamps on either side and keeps it in the closed promoter complex. Prevents it from melting because of the torsional constraints conferred by the double clamping. That is a very sophisticated way. In fact, just think about that. Why would that be? Why did it have to get so complicated? Well, the reason is you have preloaded that promoter to fire immediately. As soon as one of the repressors lets go, gets a limp wrist, then that promoter's already there ready to take off. It ensures you get immediately quick exploitation of a nutrient. Maybe you can beat your rival bacteria. And here you see the C terminus, the 4-member leucine zipper. Here you see the 2 monomers. Here you see a close-up here. This is one DNA binding domain, helix turn helix. What is that? That's the hinge helix. Notice it's in the minor groove. This is the major groove. This is where the helix turn helix is, helix 1, 2, 3, and then helix 4 is the hinge helix. And notice helix 4 is being shared with the hinge helix from the second monomer. This is the 2 core domains that hold the 2 dimers together. This is one dimer. This is the second dimer, and they're held together at their C-terminal tails by a 4-member leucine zipper. And this is more of the same. That's the hinge helix, or helix 4. The IPT binding site is right here. This region here, the 2 core domains hold the 2 monomers together to form a dimer. This is what a helix turn helix actually looks like up close. As you see, it's really a triple helical cluster reinforced by beta strands. That's the full text here. It's triple helical cluster reinforced with beta strands. It's really more complicated than simply helix turn helix. No, it's got another helix in the background here and a couple of beta strands to form a little small sheet that gives it structural integrity. Okay. Okay, the main thing you need to get from here is how this thing really works. How does binding IPTG cause it to go to release? I talked about the hinge helix getting kicked out of the minor groove, but let's think about it in a little more detail. You can think of the 2 monomers as joining together like this. And each, at my fist, that's the DNA binding domain. The helix turn helix, helix turn helix. And they together are binding a single operator. If you bind the inducer, which is right here in the middle, it causes the 2 domains to rotate away from each other. And when that happens, the spacing is all wrong. It can't bind that inverted repeat anymore. It can bind one at a time, but they're too far apart for it to bind the 2 inverted repeats simultaneously. So you see, moving them apart makes it so it no longer fits onto the operator. That mechanism of binding a small control molecule and causing the dimer DNA binding domains to rotate away from each other is also used by CAP. But in the case of CAP, when they move away from each other by binding cyclic AMP, now they fit perfectly. Same mechanism, 2 opposite outcomes. And that's just what this says. What happens to LAC repressor when it binds the inducer molecule? 2 dimers separate so they can no longer bind the operator. The 2 dimers separate. I think it's the two monomers. The two monomers of the dimer separate. Yeah, that sounded more like it. So that only one at a time can bind the operator. I think I'm going with that one. The central globular domain, now I'm just filling the air with words. What happens when the central globular domains change the configuration of the hinge helix, resulting in the loss of affinity for the operator? Let's look at this one again. Two monomers of the dimer separate so that only one at a time can bind the operator. That looks right to me. I changed it. I changed it. What did I change? It was? Okay, so I did a little editing there. It looks like it should be up, what did I say before? Number two, so central globular domains change the configuration of the hinge helix. Okay. Globular domains don't change the configuration of the hinge helix. They simply move apart so they can't both bind to the operator. That, it took me all day yesterday to remove the blue. It takes 6 seconds to remove the shadows on every object and arrow and text, and then you have to remove the text shadows plus the shape shadows. That takes the whole rest of the day if you're a stupid old man. Okay, this is what you want. This is what you send off, and you order the full so you can hang it, in your room. This is the lac repressor. A dimer here, held together with the C-terminal leucine zipper, and it's binding the two operators, and it's forming a loop. That is in full repression mode, right? That's what it really looks like at the lac promoter. And there's something there, you're going, well, what's that blue mess? That turns out to be not RNA polymerase, that is cap. Oh, gets even more complicated. But then you'll see it all makes sense. Okay, when repressors binds to, loop together, the center of the loop represents cap, another regulated protein. So it turns out when the RNA polymerase is recruited to the lac promoter by the repressor, that when the repressor goes away, the polymerase would just float away, right? That's what I said, because it's such a lousy promoter. And it would if it didn't have cap to go, wait a minute, bub, you're not going anywhere. I'm going to bring you back home. The cap is required to keep the polymerase from floating away when the repressor leaves. So it is under both positive and negative control, isn't it? Cap is a positive transactivator. Repressor, of course, is negative regulation. And here's the other aspect. You would expect to see multiple operators because it's a tetramer, and what you see is there are actually 3 operators in the lac operon here. And this is the repression. If you have them all there, it's a 1300-fold repression. But as you remove various ones, you get really, really, really low repression. So it requires at least two be there. It works best if you have 3. Here you see where the operator 3 is there, and this cap, and one is sitting in front of the polymerase, and this one is into the coding region a ways. Presence of the lac repressor increases the affinity by about 100-fold. How does the lac repressor inhibit transcription? Now you know that it inhibits the transition from closed to open promoter complex. prevents the polymerase from melting the promoter. This is the leucine zipper. This is the very crude characterization of it. You have a series of leucines, which are large, bulky, hydrophobic amino acids, exclude water, but they stick together. And because of the nature of a protein helix, if you have 7 amino acids in a row, that's enough to go around something like 7.2, 7.1, 7.1 or 2 in order to go around twice. So it's not an even number. It's not like 10 goes around, you have 1 turn of the helix. It's an odd number. It's roughly 7, but it's really 7.1. So if you lined up all the hydrophobic residues, you'd end up with that hydrophobic strip on one side of the helix that would slowly curve around the outside of the helix. And that's why the 2 leucine zippers slowly turn around each other. And here's, if you look on N, you'll see that slow turn they make around each other. And this is the heptad repeat, 7, but since it's not exactly 7, it's 7.1 or 2. That's why it doesn't come out even. That's why the row of leucines are not straight up and down on the outside of the helix. So it's called a 3-4 repeat, because 3 plus 4 makes 7. So it can also be A, B, C, D, E, F, G. Positions A, B, C, D, E, F, G, and then the next one is A again. So at position A and D, these turn out to be on roughly the same helical face, but they're not lined up with each other. They're offset. So you have a strip of 2 hydrophobic residues going up the side of the helix and slowly turning. As you see here, position A and D don't line up over each other, so you have actually a double row of hydrophobicity. Leucines here, leucines at D. Usually D has the most leucines, but A and D are where the hydrophobicity lies, and so that provides a face for the 2 helices to interact. Okay, so we talked about the lac repressor, and I'm always confusing, not in my head, but when I speak, lac and lambda. I haven't done it so far this year very much, but it turns out the lambda phage repressor interacts with RNA polymerase in a similar manner. This is represents the phage promoter, left, and operator, and this is a dimer of the repressor of the lambda phage repressor. So let's see what happens here. Notice it makes contact with the RNA polymerase, and so this is an example, another example where the repressor actually stimulates. And this is what you're going to see. Repressors, or one protein can be a repressor one day and a transactivator the next, depending on the context. In this particular, at this promoter, the left promoter, you need the repressor there in order to increase the affinity of the polymerase for the promoter. Here you see this. You don't need to memorize that at all. This is the guy who did a lot of this stuff. You're not going to need to know about him. He's probably retired now, but he's about this high. Well, yeah, good thing. And he plays a Stradivarius violin. He's fabulously wealthy. He got it from biotech somehow. He was a Harvard, at the Rockefeller. And he'd play out there on the lawn at lunch, and people eat their little sandwiches with no crust or something. And he plays golf in the Caymans and stuff like that, but he's long retired. But he was really a great, worked out the structures of this, the helix, turn helix. Okay. Lambda activates by contacting sigma 70. So here you see the lambda phage, not lac, we've jumped. The phage repressor, when I said it made contact with RNA polymerase, it's making contact with one subunit, our favorite, the sigma. It's stabilizing sigma to bind where there's a very lousy minus 35. That strategy is also employed by CAP. You see the common themes emerging here. Because this is CAP. It is right next to domain 4. You remember domain 4.2 binds to minus 35? There's interaction with CAP with sigma. Lambda repressor makes contact with the same general region of sigma to when the repressor acts as an activator. Okay, so we've covered the structure of the lac operon. How much time is it? Got plenty? Whoa, that's a dangerous thing to tell me. We covered the structure of the lac operon. The lac repressor, we know all its structure, all its little domains. We know it binds as a tetramer and has to grab hold at two sites. And so in the repressed condition, if I'm presenting you with diagrams of promoters, which I will do, I'll ask you to say, look at that promoter and tell me whether the gene is on or off. So what are you looking for? If you see a big DNA loop, you know that gene that must be off, right? That means that the lac repressor as a tetramer has bound at two sites and is making a loop. The loop is the shutdown configuration. Okay. We'll learn more, we'll learn to think about when it's on and off in just a minute, but we're going to introduce a second operon now. It's also involved in breakdown of a sugar, arabinose. And it also has a DNA looping mechanism as part of its regulation. And just like the lac operon, when it's in the loop, it's not active. Except this forms a loop by an entirely different mechanism. Not entirely, but a different twist to it. So here we see the BAD, arabinose, BAD, these stand for the cistrons, the arabinose operon, and notice it has two the operon itself is right here, and this is a gene close by that encodes the repressor. And this is a clear example, and the repressor is this arabinose C protein. We'll also find out that this arabinose C protein, depending on whether the inducer molecule is present, it changes from a repressor to an activator, same protein. This happens. You just can't prevent it. It does it. Let's see how all this works. Well, look at this loop here. If you saw it in that loop configuration, this loop doesn't prevent formation of the open or melted promoter directly. This actually, the DNA that's dangling off the end of this loop just gets in the way of RNA polymerase binding to the promoter. Instead of having a protein get in the way, this has bent a piece of DNA, and the DNA now is getting in the way physically of polymerase accessing the promoter. It's kind of a novel way to use a loop. If you could just undo this loop, then the polymerase could have access to the promoter. Except it's a lousy promoter. You knew there'd got to be some complication. This promoter requires cap. The lac promoter requires cap. This one does too. What's all this requiring cap for? These are both genes involved in degrading sugars, and they both are regulated positively and negatively. And cap, the same protein, is always the positive component of their regulation. What's going on? It turns out E. coli is a glucose specialist. If there's any glucose in the gut or wherever E. coli finds itself, E. coli has evolved to drop everything else and be the first guy at the table with the napkin tucked in to eat the glucose. E. coli's evolutionary strategy is, if there's glucose around, I'm going to eat all of it before anybody else can. I'm not going to waste my time degrading arabinose, degrading lactose. No, no, no. I'm a glucoseaholic. And that's why, if glucose is there, you want a way to shut down all those other sugar degrading processes, so its full attention can be devoted to degrading the glucose only. And that is catabolite repression. All those other pathways are shut down in the presence of glucose, and that's regulated by cap. We'll see how that works in a minute. So it turns out that if glucose is present, then, see, how does that work? If glucose is present, then those others are going to be shut down, so you're going to have you're going to have cap binding. I'll get to that in a minute. Glucose inhibits the enzyme that makes adenyl cyclase. Inhibits adenyl cyclase, which degrades the cyclic AMP. Is that right? I've got it backwards? It'll come to me as we get there. Let's look at the conditions. So you have a cap binding site, and you have two places where the arabinose C protein can bind. One has an O, which means an operator, and the other has an I, which means inducer. When the arabinose C protein is acting to repress, it is bound to operator by definition. But when it's only binding to induce, it's the same sequence. It's just location. It now is called an inducer binding site. So let's look. Arabinose C without the presence of the signal molecule, the substrate, arabinose, it actually is like a circus act. One stands on the shoulders of the other one, and it forms the loop. Its feet are down here in inducer 1 site, but the other one is bound to the operator, operator 2, it turns out. That forms the loop. If you have arabinose present, it causes the one on top to lose affinity for the operator and gain affinity for inducer site 2. So the arabinose C protein is always bound to the promoter. It just depends on whether it's doing a circus act or not, whether it's forming a loop. If arabinose is present, represented by that diamond, they're bound side by side as a dimer on the 2 inducer sites. The loop goes away. This is the operator 2, which is here. This thing unfolds. This is where cap is binding, and cap plus arabinose C allows transcription to proceed. Okay. Catabolite repression, cap is 200 base upstream, positive regulated bounce, okay. It can be best understood by looking at that picture. And let's see here what this is. this is just another representation. That's the two little guys one standing on the shoulder of the other one to form a DNA loop. In the presence of inducer, they no longer do that. If you add or remove 5 base pairs between those two sites, between O2 and the first binding site, then that's enough to mess up the interaction between the head to shoulder dimer. So the helical face is very important. Otherwise you'd have one guy binding, but his partner wouldn't be underneath him. So you have to have them binding on the appropriate helical face so when they form the loop there won't be any tension. Okay, there's more regulation. Let's look at, okay, right here. So we talked about O2 forming a loop with the inducer site here, but we didn't talk about operator 1. Let's go back and look where operator 1 is. Operator 1 doesn't seem to be doing anything, right? And we've got this other promoter. That promoter is going that way. That is the promoter that is making arabinose C protein itself. This operator is repressing because it's sitting in front of the promoter that makes the arabinose C protein. This is what makes the degradation polycistronic message right here. This is the one recognized on the right, being recognized by the DNA looping. So this promoter, see, going the other way, is actually encoding. I had this in the wrong, I said it was here, it's past it. The polymerase goes all the way down here, but the Shando-Garno is right here, and you start translating the arabinose C protein here. If you have arabinose C binding as a dimer at operator 1, then it blocks the RNA polymerase. So this is auto-feedback. As the concentration of arabinose C gets lower and lower, then there's not enough to fully occupy this operator, and if there's not, then the polymerase can go all the way down here, make a messenger RNA, which is then translated to make more. So it auto-regulates itself to keep the levels steady. That is typically how repressors work. We have taken 2 screwball examples to show you how sophisticated it can get. The lac operon had this crazy DNA loop tetramer configuration. Arabinose has all of the above. It has the arabinose C protein acting as a positive acting transcription factor, and as a repressor, auto-regulating in a feedback mechanism its own synthesis. Whew. What's the time? We got 25 minutes? Then I should keep going. Are there any questions on this? Does this seem clear, or is this going to take some more study? Take some more study. You kidding me? But some of you, it's sort of familiar, and others, it's going to have to go back and review it, right? Are there any things that you want me to say over again, explain right now before we go to the next one, which is CAP? We've been talking about CAP. I'll just go in more detail in CAP. Yeah.
I'm confused on the lactobacillus protein. Did you say that the arms, do they bind the same operator or 2 different operators?
Okay, good question. Each monomer binds to 1/2 of an operator. You know, the operator is 2 imperfect inverted repeats. And so each monomer of the dimer binds one of those little arrows. Another question?
For the arabinose repressor, does it have different binding sites? Because in the first figure you showed it binding to the RI sites and the operator, and then in the second figure you showed it binding to 2 operators, and then the third you showed it.
Okay, you're talking about the lac?
No, the
Arabinose, there are actually 2 arabinose operons, which we'll learn in the next PowerPoint. There's the BAD polycistronic operon, and there's the FGH, and each has sites for binding arabinose C. Okay, so you understand the concept of a protein can act both as an activator and repressor simultaneously. The lambda repressor is an example. It represses on, there are 2 promoters in the lambda phage, right and left. On the left, it's actually attracts, it needs to make contact with sigma to stabilize sigma's binding to the missing minus 35. On the right promoter, which I didn't show, it's acting as a classical repressor. It's getting in the way of RNA polymerase. That promoter is usually off. So that's the lambda phage repressor performing 2 roles to activate and repress. In a sense, the lac repressor does that because it attracts and stabilizes the binding of RNA polymerase to the lac promoter by being underneath and making contact. So it pre-primes or primes the lac promoter for rapid activation. And then, of course, arabinose C is just the most blatant example we've covered, where it's always bound. It depends on whether it's head to shoulders, forming a loop, or whether it's side by side, which now it becomes required and acts like a positive transactivator. In fact, it acts as if it were cap. Makes the same contact with the polymerase in sigma that stimulates transcription. Okay, let's go to the next one. We definitely will not get through this one, but it's, I guess we won't, we might. Catabolite repression. I've already begun to talk about this. When this, you see here a dimer of CAP. CAP is cyclic AMP binding protein. CA is cyclic AMP protein, CAP. Binds as a dimer, and in the case of CAP binding, it's long been noted that it really bends the DNA where it binds. And everybody's tried to think of a reason why that might be so. Does it help melt the promoter? Doesn't seem like it does, but it bends it more than 90 degrees. Some reports say 130 degrees. Really puts a serious kink. Of course, it's a helix-turn helix, a helix-turn helix, and the helix-turn helix. And right in between the domain that holds the 2 monomers together, guess what? It's a leucine zipper, but this is a 2-member leucine zipper. Cyclic ATP binding site is there in the red. This is from your textbook, and so I added this year. And you see it really bends the DNA. This is upstream, and so they speculate as to why this may be, so I just kind of drew in what I thought. Why not? If you're trying to make contact on either side of CAP, which is what all the little diagrams say happened from footprint data, one of them you have to reach way around. But if you bent the DNA, the distance that the linkers have to go is about the same. Maybe that is why the DNA is bent so fiercely. I don't know. Maybe so. Give me a better story. Anyway, it really puts a bend and a big kink in the DNA. Notice how this has similarities to the lac repressor. You have, this is a dimer. It's one dimer, not two dimers. But in the middle you have a site for binding cyclic AMP, and guess what? When it binds cyclic AMP, those two DNA binding domains separate, just like the two monomers in the lac repressor. They separate from each other, except when they separate, in this case, they now fit perfectly to the binding site. Same mechanism, opposite effect. Here you see these two domains labeled C. These are really the leucine zippers. That's where cyclic AMP binds, and this is the helix-turn helix, the DNA binding domains. Okay, location is important with the CAP activator. What you'll see is that location is very important. There are three configurations, and you'll need to be familiar with them. One is there is a binding site at -41.5. If it binds there, you know it's a certain type of mechanism in play. 41.5. Where's the -35? It -35, right? And so it's going to be right beside the -35. Whenever CAP binds there like that, it compensates for the fact that it doesn't really have a -35. It is there to touch domain 4 of sigma and calm it down and say, it's okay, there's no -35, but I'm here to keep you in place. CAP is required because it has such a lousy, poor baby, -35. It's a positive transcription factor. It's required. It's a deliberate flaw in the promoter so that it can be regulated. Then there are these other configurations. -61. It turns out every 10 bases you could have a binding site, roughly, which means the helical face is important. Takes 10 to go around, so it has to be on the same helical face. But other than that, it can go all the way out to about -91 or so. And if it's in this kind of configuration, well, it can be there also, but if it doesn't have a -41.5, you know it's either one of these other two, and we'll learn more about those in a minute. The distances must be an integral number of DNA turns, which means multimers of 10. It must be on the same helical face of DNA. No CAP-dependent promoter has a very good -35. This is the flaw that CAP compensates for. That's how it can regulate. In the GAL promoter, CAP increases the transition from closed to open. CAP, in some configurations here, which we'll learn about in a minute, CAP can actually stabilize sigma and allow sigma to melt. And that's this configuration right here. When it's at -41.5, it is making direct contact with sigma. If sigma didn't have that direct contact because it has such a lousy -35, sigma would be unable to melt the promoter open. The GAL promoter happens to have this configuration. More about that in a minute. In the LAC promoter, CAP increases the formation of the closed promoter complex. It's another way of saying it has other places where CAP binds. It just doesn't bind to that really, really close site where it can make direct contact with sigma. Fun facts. Yeah, right. CAP controls more than 100 promoters. This glucose thing, CAP is a servant of the glucose levels. 100 promoters, relatively small protein, binds to the dimer. It's the first transcriptional activator in any living system to be characterized and get 3D structure. Simplest cap promoters, 3 components, cap, RNA polymerase, and DNA. Most are more complicated. We'll appreciate that in a minute. So what we have now are the 3 classes of cap-dependent promoters, and that's pretty much it with a few other added information. So if you can remember these 3 things, then we're through with cap, and all we have is this attenuation thing, which I'll have the audio file. I'll do an audio file on this also. Class 1. There's going to be class 1, 2, and 3. Class 1 requires only cap. It doesn't require some other positive actor, transcription factor, just cap. And there's only a single site for cap binding, and it can be at various distances, although they must be on the same helical face. This is the one where they're bound at minus 61, 71, 91. This is not the one where they're bound at minus 41 and a half. You need to remember the example of that, which is the lac. Here you see, could be minus 71, 61, 72, 91, the same helical face. It is not located here at minus 41 and a half. That would make it a class 2. So class 1 only needs 1 cap dimer, and it is located at these special positions on the same helical face, but it is not located super close. An example is the lac promoter. It makes contact with the alpha CTD, and here you see the DNA binding domain itself. It's close to the binding site for cap, is actually making contact with the alpha CTD, and that is how it activates. It doesn't activate by stimulating sigma. It activates by increasing the affinity of alpha for the promoter because it doesn't have a UP element. Alpha has to make contact with something else, and it makes contact with cap. That's its mechanism. That's how it stimulates. Here you see the structure again of the cap dimer. The activator, response site 1. This is the contact in the DNA binding domain that actually makes contact with the alpha CTD. There are 2 of these regions on cap that are needed for activation. I think it's activation region 1. The other one's up here. This is the one that's in the DNA binding domain and makes direct contact with the CTD of alpha. You got 2 alphas, you only need to make contact with one of those. I need to change that to alpha symbol instead of an A. Class 1, you can see it here. Mechanism, interaction between cap and increases the binding constant for formation of the closed promoter. There's nothing here about facilitating melting. This just enhances the initial interaction of the polymerase with the promoter, keeps it from floating away because it's tethered to cap. And it makes contact with AR1. Now let's go to class 2. See, we've only got one more we've done, right? Class 2, again, it only needs 1 cap dimer. Why do I keep saying that? Because class 3 is going to require 2 of something, one of which has to be a cap dimer. So class 1 and 2 require 1 cap dimer. It's just a question of where it's located. This is the one that's always located right there at minus 41 and a half. Period. It's a class 2. And this is the one where it makes multiple contacts. It's making contact with the CTD and behind here, it's actually making contact with the NTD of alpha again. And it's stabilizing sigma all at the same time. The gal P1 is an example. When it's sitting in jammed up like this at minus 41.5, it is necessary for sigma to melt. So you have to have this class 2 configuration before it's involved in facilitating the transition from closed to open promoter. And notice where the CTD is binding. It's like reaching all the way around like at the movie theater, way on the other side. Oh baby. Ain't this comfy? And there's a second site for interaction. This is AR1 on cap. That is activation region 2 making contact with the N-terminal domain of alpha. Of course, there's just no minus 35 there at all. What's the difference between the class 1 and class 2 cap-dependent promoters? Let's see, location of the cap binding site. That sounds good, but I miss these all the time. Location of the minus 35, no, it doesn't really have a minus 35. Number of cap dimers, no, they both require 1. Type of sigma used, no, it's got to be 1. Okay, I threw this in just to make life really, really cool. This is demonstrating how, this is one of the proofs that demonstrated that cap was required to melt the DNA by sigma. It's kind of a roundabout way to do it, but what they did is rifampicin, which we've talked about before, and they take cyclic AMP, which is required for cap binding. Cap can't bind except cyclic AMP is present. So you have cyclic AMP in both configurations, and now you've just added cap. You've got the polymerase, and then you get it to bind first, and then you add the antibiotic and nucleotide triphosphates simultaneously and see whether transcription works. And what you see is there's no transcription if there is no cap. And that's because that rifampicin cannot get into the interior of the enzyme if it's already gone into the open promoter complex. So that's why rifampicin has no inhibitory effect down here. You can open the promoter complex, and once it's open, then you add rifampicin. Rifampicin can't get in here, so transcription is not inhibited. The take home was, and I won't give you this figure on the exam, the thing to remember is that at GAL promoters, the cap binding is involved in facilitating the melting by sigma of the promoter. It's a class 2 cap dependent promoter. That's the one where it's jammed in at minus 41.5. Class 2, here you go, this is that second activation region. You know, it can bind, should be a prime, it didn't come through on this computer. The cap makes contact, there are 2 alpha subunits, right? I said one makes contact with beta and the other one with beta prime of the core. You will not need to know this detail on the exam. Cap is able to stimulate one of those a little better than the other one. Not something you need to know. Class 2, here you go again. Downstream cap, the one that's behind here, this is the downstream cap and the upstream cap. This one is the one that's making contact with the N-terminal domain of alpha. The upstream cap monomer is making contact with the DNA binding domain, or the AR1 of the CTD. That's the AR1's in cap that's making contact with the CTD through AR1. Finally, class 3. Oh yeah. Class 3 is different. It's a cap-dependent promoter, so cap is involved. But now you need 2 cap dimers. Or you have to have two of something. One of which, though, has to be a cap dimer, right? Can you think of a class 3 that we've covered? Arabinose, right? It required cap and arabinose C. Two things, one of which is cap. It could be two places to bind cap dimer. It would still be a class 3 cap dependent promoter. And there are lots of configurations here that you could have, but notice the common thing here is you have two caps. Here you have a combination of a class 1 and a class 2, but because it has two, you automatically throw it into this sort of catch-all category called class 3. But you look at it, it's really a composite of a class 1 mechanism and a class 2, because it's located right there at minus 41.5. And notice what we got down here. We have a way that you can inactivate an activator. If you have a protein CYTR, it turns out if you have a class 3 promoter and you have a CYTR site and you bind this dimer in between the two caps, you can block the interaction of cap with the CTD of alpha. So this is called anti-activation. I guess you could have anti-activation. So you could regulate whether or not cap is going to work by controlling the presence or absence of a third or second regulatory protein. May use class 1 mechanism, or could use class 2, a combination. Here you see, can use both at the same time. What's important is it needs two regulatory sites. Both can be cap, or one can be cap, or arabinose C, or whatever. Sometimes class 3 has an additional layer of regulation called anti-activation. And here you see an example of this. The CYTR protein can destroy the ability of cap to activate. In fact, there are other kinds of anti-activation mechanisms, but they only apply to class 3 promoters. Some bacteriophage block activity of the polymerase. Wait a minute, block activity of E. coli polymerase. T4 blocks cap interaction by ADP ribosylation. So this that doesn't seem like a pure play, does it? You just inactivate the host RNA polymerase. But this one certainly is an anti-activation. This T4 phage modifies the CTD so it can no longer, by ADP ribosylation, so it no longer can interact with cap. So you have a lot of the several anti-activation mechanisms. How do class 3 cap-dependent promoters differ from class 1 and 2? Class 3 always require cap plus another type of activator. Class 3 require cap dimer, whereas classes 1 and my monomer, class 3 requires two activators, at least one of which must be cap. Yeah. How does that differ from this one? Cap, class 3 always require cap plus another type.
That one says it's always cap and one other thing.
And one other
Sometimes it might be two cap.
require two, at least one of which must be cap. This says always require cap, plus another. This says it can't be two of the same. I am so tricky. I would never get through this course. It's got to be three, right? At least one of which must be cap. You don't have to have, it doesn't require that one be cap and one be another. They can both be cap. Whew, got it right. Okay, last stretch here. This is another representation of the BAD, the promoter we've been talking about. You remember, this is now how they have depicted arabinose C. You have a DNA binding domain on the bottom, and then you have a oligomerization domain at the top. Here, notice that arabinose C is binding in front of cap. The CTD of alpha is stabilized by making simultaneous contact both with arabinose C and with cap. Arabinose C is a mimic of cap. They're interchangeable. The second promoter, which I told you, there's another operon regulated by arabinose C called the FGH, promoter FGH. And notice the configuration is switched. Cap is sitting at the minus 41 site, and arabinose C is sitting at the minus 61. Here, cap at minus 61, arabinose C jammed up at the minus 41 and a half, as if it was cap. And the dead end promoter, and I think this is it. At the FGH, there's a really strong binding site for RNA polymerase. It is very seductive, though. It binds it, but never lets it go. So it wants to bind here. If it binds here, it never can melt the promoter. It just stays stuck here. It's called the dead end promoter. But if it has arabinose C and cap bound at their sites, then the attraction to the CTD is enough to pull back, to tether the polymerase so it doesn't go too far and gets positioned at the productive promoter. This is what you need to know, how to predict activation state of lac and arabinose operons. Okay, how much time do we have? I'm out? Okay. What comes next is very important. But I don't have time to, if anybody wants to stay, I can explain it, but I'll do an audio file to explain it. What I'm going to do is on the exam, show you a configuration of a promoter. And I'm going to ask you, what's in the media? Uh-oh, which way is north? There's not enough information there, right? I'm going to show you whether cap is there or not, whether a repressor is there or not, whether the DNA is looped or not. And I'm going to tell you how to think about that in a stepwise fashion so you can tell me whether in the media, I'll ask you two questions. In the media, do you have any glucose? That's the first thing and if you looked at that picture, what would you be looking for? What is your clue for whether you have glucose or not? Cap. Cap is regulated by glucose. If you have glucose present, then you are going to have, I've got it wrong in my head. I know it should be glucose yes, cap no. Glucose inhibits the enzyme that makes cycling. Okay, yeah, that's right. If you have glucose, you cannot have cap because glucose inhibits the enzyme that makes cyclic AMP. If you don't have cap, all of those other sugar operons can't work, right? They require cap. That's how glucose regulates and controls all those other pathways. It inhibits the enzyme that makes cyclic AMP. So cap can't bind to any of those others, and all the rest of them require cap. So if you saw cap there, you'd go, well, if cap's there, there can't be any glucose. That's easy. Just look at CAP. CAP is your signal. CAP is there, no glucose. Okay, the next question I'll ask is the particular sugar present? I'll only ask you about arabinose and lactose. So I'll say, is lactose present? Or I'll say, is arabinose present? What would you look for then? Well, in the case of the lac operon, you look for DNA looping would be a real easy one, right? The repressor would let loose and the DNA loop would go away. So if the loop is gone, you know you've been messing with the repressor, you've got de-repression going on. There must be lactose or beta-galactoside there. How about in the arabinose? Same thing. If the loop is there, there's no arabinose. So the DNA loop means in both cases you do not have the inducing sugar. So you could tell me whether glucose is there or arabinose is there just by looking at it. Just in a moment you could tell me, is CAP there? CAP's there, no glucose. Is there a loop? Then there can't be an inducing sugar. If there's no loop, yeah, there could be lactose or arabinose, depending on which operon you're looking at. But would that, if the sugar is there and there's no DNA loop, would transcription happen? Nope. Not unless CAP was also present. It requires two things, that the loop go away and CAP bind. That's kind of a trick. You know, oh, there's no loop, must be active. No, look carefully, is CAP there? And that's about as mean as I can get on the exam. And other courses you probably had big charts where you have to fill out and figure it out. CAP and loop. You don't have to do that here, just understand how it works and you can handle any question I can ask you. I'm going to show you what the picture of the promoter, I'm going to ask you, is glucose present and is arabinose present or lactose? And be easy to give me that answer. And that's what the rest of these slides talk about. That's it. So all you need to know now, what's left, is the audio file on how that anti-terminator thing works with all the hairpins. That'll take about 30 minutes. It's a short audio file and you can figure that out once you see me explain it, hopefully.